Good morning, ninth class students. Welcome to our incredible biology class. So, we have started with first chapter of our biology, the fundamental unit of life. And so far, we have discussed what is a cell, discovery of cell, cell theory, and type of unicellular and multicellular organisms, prokaryotic and eukaryotic cell. So, let's start with structure of cell. So, a typical cell has three parts. Be it a prokaryotic or a eukaryotic cell, it must have three parts. The covering of the cell, that is the cell membrane or plasma membrane, the genetic material of the cell, and the last part is the cytoplasm. Okay, these are the three basic parts of a cell. So, let's start with the first part of our cell, that is plasma membrane or cell membrane. So, we have discussed some functions of plasma membrane or cell membrane. Let's quickly revise those. So, what is a cell membrane or plasma membrane? Cell membrane is the outermost covering in animal cell and inner covering in plant cell. So, the plasma membrane or cell membrane maintains shape of the cell. Okay, it maintains shape of the cell, it provides shape to the cell. Now, the second function of plasma membrane is it protects internal content of the cell from external environment. Plasma membrane is selectively permeable. What is the meaning of selectively permeable? Permeable means which allow entry and exit of substance. And selectively permeable means it allow entry and exit of some selected substance. So plasma membrane is selectively permeable which allow entry of solvent and some selected solute. What is solvent? What is solute? Let's take one example. If we are dissolving a spoon of sugar in water, now sugar is solute and water is solvent. So plasma membrane allow entry of solvent and some selected solutes. So plasma membrane is made up of bilipid layer, two layer of lipids. Lipids are fat. So two layer of lipids are present in plasma membrane, and in this lipid layer proteins are embedded. Okay, plasma membrane also consists of carbohydrates which are attached with the lipid layer. Okay, so the structure of plasma membrane was proposed by Singer and Nicholson in 1972. The structure of plasma membrane was proposed by Singer and Nicholson in 1972. So they proposed fluid mosaic model of plasma membrane. The fluid word represents lipids and mosaic word is basically used for the pattern which is formed by proteins embedded in the lipid layer. So fluid mosaic model is also known as protein iceberg floating in the sea of lipid. Because we know in sea icebergs float. The same way the sea is made by lipids in the plasma membrane and protein act as the icebergs which float in the sea of lipids. So icebergs can change their position because they are moving. The same way proteins in the lipid layer are also floating and they can change their position. Now according to Singer and Nicholson, the thickness of plasma membrane is 7 nanometer or 75 Armstrong. Okay, the thickness is 7 nanometer, or we can also call it in 75 Armstrong. Okay, so this is the fluid mosaic model which was proposed by Singer and Nicholson. Please have a look. I have a simplified diagram for you. Now you can see two layers are there represented by blue and black color. So blue spherical color represent head of the lipid molecule while black color represent tail of the lipid molecule. Now the nature of the head is hydrophilic. The word hydro means water and philic means loving. So this head is hydrophilic which loves water. So it is in contact with the water. So head is always present on inner surface as well as on outer surface. While tail it points inward. Tail is pointing inward. It is hydrophobic. Phobia means fear. Okay, So it hates water, that is why it is pointing inward. Head is present on outer and inner surface while tail points inward. Now, two types of protein are embedded in lipid layer. One protein I have marked with this magenta color. This is the peripheral protein. Peripheral protein are also known as extrinsic or external proteins. They are also known as extrinsic or external proteins, which are partially embedded in the lipid layer. Okay, they are in contact with one of the layer. They are not fully embedded, they are partially embedded, half inside, half outside. Okay, now the another proteins marked with light pink color is integral protein, or also known as intrinsic proteins. They are fully embedded in the bilipid layer. They are fully embedded, they are in contact with the both the lipid layer. Okay, and glycoprotein are also marked here with the green color. Glyco word represents carbohydrate, so carbohydrate chain is also present here. So from this diagram we can conclude that plasma membrane is made up of phospholipids, phosphate group, and lipids. Lipids are fats, and proteins. So basically there are two type of protein, intrinsic and extrinsic proteins. Okay, the integral protein, they act as channels for entry and exit of substance. Okay, so here is a trick to remember this fluid mosaic model. Let me revise you. Basically there are two layer of lipids, and lipid molecule is made up of head and tail. Head is hydrophilic in nature, which loves water and which is in contact with the water, while tail is hydrophobic, which hates water, so it always points inward. So how many layers are there in the plasma membrane? Two layers are there in the plasma membrane. Okay, this the same way when we eat sandwich, so what is present in the sandwich? In sandwich, two layer of bread are present. The same way in fluid mosaic model, two layer of lipid is present. Now what is embedded in the sandwich? Cucumber is there, onion is there, tomatoes are there. Okay, the same way basically there are two types of protein embedded in the lipid layer: intrinsic protein, extrinsic protein. And lipid molecules also have chains of carbohydrates. Lipid molecules also have chain of carbohydrates. Okay, so you can learn the topic from, you can learn the structure of plasma membrane through this trick. And this diagram, this simplified diagram, you have to draw in your notebooks. Okay, in your fair notebooks, draw this fluid mosaic model. Now as we are discussing that through the plasma membrane substances are moving. So basically there are two types of transport through the plasma membrane: active transport and passive transport. Now what is the meaning of active transport? Active transport is that type of transport which is taking place by using energy, by utilizing energy. And in active transport, substance move from low concentration to high concentration, from low amount to high amount. Okay, what is passive? Passive is opposite of active. In passive transport, substance move from high concentration to low concentration without using energy. Okay, in active, substance move from low to high by using energy, while in passive, substance move from high to low without using energy. Now what is a bulk transport? In bulk, what is the meaning of bulk? Bulk means large. So in bulk transport, large amount of substance move across the plasma membrane. Okay, so endoosmosis, endocytosis, the method by which cell is taking its food, endocytosis, phagocytosis. Okay, they are the example of active transport. So this is the figure to explain active and passive transport. Okay, we are having a cycle and we have to go up to the hill. Now we are moving from low to high. So what we have to do for riding our cycle, we have to apply energy. Okay, we are using our energy and we are riding our bicycle. So when we are moving from low to high with the use of energy, it is active transport. Now we reach at the top of the hill, we have to come down. So now also does we have to use our energy? No. Okay, we will just simply sit on the bicycle and we are moving from high to low. So without using our energy, we will come from high to low, from uphill to downhill. So this is the passive transport. So when we are moving from low concentration to high concentration with the use of energy, it is a type of active transport. And when substances are moving from high concentration to low concentration without using energy, that is the type of passive transport. I hope active and passive is clear through this diagram. Now, diffusion and osmosis are the two types of passive transport. So, in passive transport, we have already studied the definition of passive transport. One substance moves from high concentration to low concentration without using energy. Now, then what is the difference? The difference is osmosis requires a semi-permeable membrane. Semi-permeable membrane which allow entry and exit of solvent, and our plasma membrane allow entry and exit of solvent, so it is semi-permeable. So when substances are moving from high concentration to low concentration without using energy through a semi-permeable membrane, it is example of osmosis. And in diffusion, simply when substances are moving from high concentration to low concentration without using energy, without involving semi-permeable membrane, because it does not require any semi-permeable membrane, it is the example of diffusion. Now diffusion can take place in solid, liquid and gases. So let's take one example of solid. Naphthalene balls which are solid, when they are kept for some time, their size decrease because that is diffusing into the air. Okay, example of liquid, alcohol. Alcohol is highly volatile, so it is a liquid and it will easily diffuse in the air. And gases, carbon dioxide and oxygen is moving in our body, so the exchange of gases which is taking place inside our body between the blood and the cell is diffusion of gases. So diffusion takes place in solid, liquid and gas, while osmosis only takes place in liquid medium. For example, the roots of the plant are absorbing water, so they are absorbing through the process of osmosis. Okay. Now next is diffusion diffusion does not require semi-permeable membrane, osmosis requires semi-permeable membrane. In diffusion, equilibrium is attained. What is the meaning of equilibrium? If we light an incense stick in one corner of the room, the aroma will spread in the complete room. Okay, so that means the particles are diffusing equally. Okay, so in diffusion equilibrium is attained, while in osmosis such equalization is not possible. Okay, in diffusion substance diffusion independent of one another. Okay, and in osmosis only solvent undergo diffusion, as we have already discussed. It is the movement of liquids, it is a movement of water. Okay, diffusion is not influenced by the presence of any other substance, while osmosis is influenced by the diffusion of solutes. Okay, the more solutes are present, the more concentrated is the solvent. Okay. Now what are the type of osmosis? Basically there are two type of osmosis, endoosmosis and exoosmosis. What is the meaning of endo? Endo means inside. So whenever water is moving inside, water is entering the cell, it is endoosmosis. And what is exoosmosis? When water is moving outside from the cell, it is exoosmosis. Okay, there are many examples of endoosmosis and exoosmosis. Endoosmosis, when we soak raisins, almond in water, they swell up, they become turgid because water is entering into them. Okay, when we submerge our fingers for some time in water, our fingers get wrinkles because water enter into our skin and it will it become bulge. Okay, so these wrinkles are because of endoosmosis. Now exoosmosis, when we dry something in the sunlight, okay, we dry raw mangoes for the pickle, so it is an example of exoosmosis. Whenever your mother cook vegetable, when she put salt on the vegetable, ask her what happens. The vegetable will lose water, so it is an example of exoosmosis. Okay, now we will see what will happen to the cell if we place a cell in different types of osmotic solution. Okay, now basically there are three type of osmotic solution, students. Number one is hypotonic solution. Hypotonic solution which has osmotic concentration lower than another solution. Yeah, we can say that hypotonic solution is that solution which has more water and less solute, which has more solvent and less solute. If we put a cell in such a solution which has more water compared to the cell, then what will happen? The cell will gain water, endoosmosis will occur, and the cell will become turgid, it will swell up. For example, when we put raisins or almonds in the water, they will absorb water, endoosmosis will occur because water act as hypotonic solution to them. Okay, so when we put a cell in hypotonic solution, they swell up and the cell become turgid. Now the next solution is isotonic solution. What is the meaning of iso? Iso means equal. So isotonic solution is that solution which has an osmotic concentration similar to another solution. Okay, now if we put a cell in isotonic solution, now the solution has same osmotic concentration that of the cell. The same amount of water will enter the cell and the same amount of water will exit the cell, so the cell shape will remain same. So this type of cell is known as flaccid cell. Nothing will happen to the shape of the cell, it will remain same. So such type of cells are known as flaccid cell. When a cell is placed in hypotonic solution, they will swell up and the cell is known as turgid cell. When a cell is placed in isotonic solution, okay, now the cell shape does not change and it will remain same, so it is known as a flaccid cell. Now what is hypertonic solution? Hypertonic solution is just opposite of hypotonic. Hypertonic solution has osmotic concentration higher than that of another solution. For example, in hypertonic solution they have more amount of solutes and less amount of solvent. For example, if we are putting 20 spoon of salt in a glass of water, it will act as a hypertonic solution. Okay. Now what will happen if we placed a cell in hypertonic solution? Now more amount of water is present inside the cell and outside the cell less amount of water is present, so exoosmosis will occur. Cells will lose water and the cell will shrink. So this shrinkage of cell is known as plasmolysis and the cell is known as plasmolysed cell. Okay, so I repeat what happened in hypotonic solution. The when a cell is put in hypotonic solution, it will swell up and become turgid, and when a cell is placed in isotonic solution, it will remain same. So the cell is known as flaccid cell, and when a cell is placed in hypertonic solution, it will shrink, it will lose water, exoosmosis will take place, plasmolysis will occur, and the cell is known as plasmolysed. Okay, so this is shown with the help of diagram. You can see in isotonic solution the same amount of water is entering and exiting the cell, so cell shape remains normal. In hypotonic, cell is swelling up, cell become turgid, and in hypertonic solution, in the first figure you can see clearly the shrinkage of cell, plasmolysis of cell. So does this process remains same for plant cell as well as for animal cell? No. You can see in the diagram there is one difference. Plant cell have cell wall, while animal cell do not have cell wall, and cell wall help to maintain shape of the cell. So, in isotonic solution, red blood cells, they remain same, because isotonic solution has same osmotic concentration as compared to our cells. Okay. Now, in hypotonic solution, first they will swell up, endoosmosis will occur, they will swell up, then animal cell will burst. Why they will burst? Because they do not have a cell wall which provide rigidity. So, due to the lack of a cell wall, animal cell will burst, while a plant cell will never burst. And in hypertonic solution, both plant and animal cell will shrink, they will become plasmolyzed. Okay. Now, we have completed with the plasma membrane, we have done structure of plasma membrane, okay, transport across the plasma membrane. Now, the second part of our cell is the genetic material. So, we have already discussed the genetic material is present in the nucleus in eukaryotic cell, and in the prokaryotic cell genetic material is suspended in the cytoplasm. Okay. So, let us structure nucleus. So, nucleus is basically discovered by Robert Brown in 1831. Okay, it is very important part of the cell. Nucleus is known as brain of the cell because it controls all the activities of the cell. It is the control center of the cell. So, the shape of nucleus is spherical or oval. Okay. Some cells lack nucleus. For example, mature RBC, the red blood cells present in our body. So, mammal RBC, they do not have nucleus at maturity. Okay. The same happen with the sieve tubes. Sieve tubes are the type of cells present in the plants. They are the type of phloem, they are the part of phloem tissue. So, sieve tubes and red blood cells both lack nucleus at maturity. In plant cell, nucleus is present towards the periphery, towards the corner, while in animal cell, nucleus is present at the center. Okay. So, what are the parts of the nucleus? So, if we talk about structure of the nucleus, a nucleus have nuclear envelope. So, double membrane is there, okay, that nuclear membrane. Okay. Another in the nucleus, a nucleolus is present. We will discuss what is nucleolus. The genetic material, chromosomes are present, and the last part is nucleoplasm. The same way the cell has cytoplasm, the same way nucleus has nucleoplasm. Now, what is the nuclear membrane? Nuclear membrane is the outermost double layered membrane which enclose the nucleus or which separate nucleus with the cytoplasm. Okay, it is basically the dividing wall between the genetic material and the cytoplasm. So, nuclear membrane separate nucleus from the cytoplasm. Now, in between the nuclear membrane, pores are there, and those pores are known as nuclear pore. Okay, through the nuclear pore, exchange of material takes place between nucleus and cytoplasm. Now, inside the nucleus, a region is present which is rich in RNA, and that region is known as nucleolus. This nucleolus do not have any membrane, and this nucleolus is known as ribosomal factory of the cell. Nucleolus is rich in rRNA, so it synthesizes ribosomes. Ribosomes are synthesized in the nucleolus. Okay. Nucleoplasm is just like cytoplasm. Okay, it is a dense jelly-like fluid substance present in the nucleus. Okay, mainly it consists of water or dissolved ions, RNA molecules. Now, the thread-like structure which are present in the nucleus are known as chromatin. The thread-like structures which are present in the nucleus are known as chromatin. When these chromatin get condensed at the time of cell division, they form chromosomes, and chromosomes are the chromosomes are responsible for transmission of characters. How they are responsible for transmission of characters? Because they contain unit of heredity. Where are genes located? Genes are located on the chromosomes. Okay, chromosomes are made up of DNA. Okay, deoxyribonucleic acid. The full form of DNA is deoxyribonucleic acid. So this chromosomes are made up of DNA and proteins. Okay, the chromosomes are the genetic material. They contain genes are located on the chromosomes. Some of our body features resemble with our mother, some are resemble with our father, because we received genes from them. Okay, and these genes are located on chromosomes. So here are the different type of chromosomes on the basis of position of centromere. The spherical structure which is seen in the middle of the chromosome is the centromere, which hold the sister arms together. Okay, so centromere is marked here. In the chromosome where centromere is present at the center and both the arms are equal in size, they are known as metacentric. Okay, look at the left side, they are known as metacentric chromosomes. In metacentric chromosomes, centromere is present in the middle and both the arms are equal in size. In submetacentric chromosomes, the centromere is located slightly away from the center. So one arm is slightly long and one arm is slightly short. One chromatid arm is slightly long, another is slightly short. Such type of chromosomes are known as submetacentric. Okay. Third type is acrocentric, in which the centromere is present towards the end. So one arm is very long, another is very short. So such type of chromosomes are acrocentric. And the last type is telocentric, in which centromere is present at the terminal part. So only two arms are visible. Okay, I repeat, in metacentric, centromere is present at the center, both the sister chromatids are equal in size. In submetacentric, centromere is slightly located away from the center, so one is long, another is slightly short. Okay, and in the third type, acrocentric, it is present away from the center, so one chromatid arm, sister chromatids, they are very, one of this arm is the chromatid arm is very long, another is very short, and in telocentric it is present at the terminal end. Okay, so thank you class. In next video we will start with cytoplasm and cell organelles. Okay, revise well your lecture. Thank you.